to our educational video series exploring the most talked about and crucial topics in molecular biology. In the series, we'll tackle some of the most commonly misunderstood concepts, including the difference between RT-PCR and RT-PCR, a step-by-step guide to PCR troubleshooting, and a breakdown of the basics of qPCR, all essential knowledge for anyone working in the field. Today, let's break down qPCR, short for quantitative polymerase chain reaction, also known as real-time PCR. It is a technique used to amplify and simultaneously quantify a targeted DNA molecule. Unlike traditional PCR, qPCR monitors the reaction in real time, cycle by cycle. The basic mechanism is the same. In the first step, denaturation separates the DNA strand that is followed by primers annealing to the specific target regions, where in the last step, the DNA polymerase extends new strands. This results in exponential increase of the amplified DNA with each cycle. qPCR quantifies DNA amplification using fluorescence with two main methods, dye-based and probe-based detection. In the dye-based method, a fluorescent dye such as cybergreen or evergreen intercalates into any double-stranded DNA. With each cycle, the amount of bound dye, and thus the fluorescent signal, increases proportionally to the amount of amplified DNA. The qPCR instrument measures the fluorescence after each cycle, generating an amplification curve. Because the dye binds to any double-stranded DNA, including non-specific products like primer dimers, additional validation steps, such as melt curve analysis, are typically performed to confirm the specificity of the amplification. In probe-based method, sequence-specific oligonucleotide probes are used that contain a fluorescent reporter dye on one end of the probe and a quenching element on the other side that prevents fluorescence by absorbing the light emitted by the reporter. Probe should be located between primers in the middle of the sequence. During the PCR, the probe locates and binds downstream of the primer to the complementary target. The fluorescent reporter dye and quencher are then separated by tagged DNA polymerase that cleaves the probe. This allows for the fluorescent signal to be released, which the qPCR machine will measure and then use to quantitate the amount of DNA in the sample. In addition to the higher specificity, probe-based qPCR also saves time from analyzing the qPCR results after the reaction has finished, since you don't need to figure out whether you're looking at the correct target amplification or not. Because of the specificity of the probes, they're also useful if you don't have a lot of target material, or if you want to detect small changes in the target-like SNPs. Probe-based method can be used for multiplexing, detecting several sequences in a single reaction by using differently labeled probes that carry different reporter dyes. Whether dye or probe-based, qPCR provides sensitive, real-time quantification of nucleic acids, essential in molecular biology, diagnostics, and beyond. All that is left now is to check out our product list and choose the product most compatible with your needs. See you next time.